Hello, my students. It was really nice being able to chat with many of you the other day when we did our review session. Here is the lecture for chapter 42. As I have mentioned to some of you earlier, if you are having any problems with printing the lecture note sheets, then don't worry about it. Feel free to just download it. I always like giving out lecture note sheets whenever I can, simply because I find that it would help you expedite your work and so that you don't have to do so much copying. Although for I noticed for many of you who are quite artistic and drawing these diagrams is not an issue. Well, I hope that is the case. But in any way, if you are having trouble printing any of these out, right, don't trouble yourself with it. Then just take maybe your notebook or your blank sheets of paper to take notes. But when you do me a favor, because I need to review your lecture notes, try to follow the section headings that I have indicated on the lecture note itself. If that's an issue, then you let me know, all right? And don't forget to upload your work. All right, so this particular lecture is a lot of fun because we're going to be talking about embryonic development. So I have in store for you a few video clips that are rather short, but it actually shows the exact examples of live organisms, which sometimes is not an option in classroom. So let's take a look, all right? So this is the page 1 of the lecture note sheet. As you can see, there are quite a number of very complex diagrams, but you could also try to make your own schematic diagrams too. This is page 2, and then this is page 3. All right, so what happens after the sperm fertilizes the egg? If you take a look at this image, the egg cell actually encompasses almost all of the cytoplasm that a developing zygote is going to need. The size of this egg cell is actually millions of times larger than what a normal human cell would be. So what the sperm cell provides, it's essentially the genetic material that the future embryo needs in order to complete the fertilization process. And the moment the sperm cell and the egg cell fertilizes each other, it forms a zygote, and we call that conception. Now, some of you may even have siblings or know somebody who are twins. But do you know the difference between identical twins, which are known as monozygotic twins, or fraternal twins, also known as dizygotic twins? Now, the word mono and di in front of those two words should give you a hint right away. Mono means one, and di typically refers to two. So if you take a look at these two diagrams, do you see anything different between them? Now, if you tell me that the monozygotic twins only involves one sperm cell and one egg cell, as you see right here, then you are correct. This involves only two sex cells of different kind, and when they fertilize each other, they undergo the process of mitosis, where you form the body of the embryo. However, dizygotic twins involve the fertilization of two different egg cells, which means the woman ovulates two eggs instead of one at the same time. Sometimes this could run in a family. And then, given the millions of sperms that enter the female's body, each one of these egg cells will then be fertilized by two different sperm cells. So, take a guess. What do you think the answer to this question is? Which set of twins could be different sexes, same sex, and why? Take a moment. All right. Well, here is the answer. For monozygotic twins, they will always be the same sex. Why? Because they share the exact same genetic material, an egg cell and a sperm cell. And then for the dizygotic twins, they can actually be the same or opposite sexes because they are fertilized by two different sperm cells and two different egg cells. And so they actually can be two different genders completely by the time they are developed. Okay, so how is the body of an embryo formed after conception, right? Because if you think about it, we're only talking about having one sperm cell, one egg cell formed together. But don't forget the initial picture. Remember this? There's a very good reason why I chose to show you this. Because what essentially has to happen now is that we have to take all of this cytoplasm, all of the material that's inside this egg cell, we have to subdivide it into many different cells. So essentially, you're taking a large mass of fluid along with any of the materials inside and use it to form different body cells. But the overall size of this egg cell that you see here, it actually doesn't change shape. It's the individual cells that are formed within this cell, which later on becomes a zygote, that subdivides and becomes smaller and smaller until it reaches the normal size of a human body cell. Let me show you what I mean by that. So let's take a look. When an egg cell and a sperm cell joins together, they form a zygote, which only makes up one huge cell. This huge cell will then have to form many cells to form all of the different body parts in a growing embryo. So it undergoes a process known as cleavage, where all of the material inside this particular zygote has to be subdivided to form many different cells. Now, in a moment, I will show you how this actually happens, but you may notice the overall size of this cell, the perimeter of it, the circumference, it's actually the same across these diagrams, and that's the reason why. But the individual cells inside, not only does it undergo mitosis, forming more and more cells, so it starts off with one cell, then it becomes two, two becomes four, four becomes eight, eight then turns into 16, 32, 64, it doubles, right? And then what happens here is that they form into a large mass known as a blastula, and then that blastula then forms into a gastrula, which is what you see here with multiple layers of the body that are in primitive form. So let's go through some of the text. So one of the first stages that it produces is called morula. The morula is produced by cleavage, which is the process of mitosis, where it will form approximately 16 to 32 cells. So we're looking the staging between these 8 cells and before you reach the shape of the blastula that you see here. The blastula stage consists of a spherical layer, a round layer, that consists of approximately 128 cells. Now you notice all of these are even numbers, right? Because in mitotic cell division, every division produces 2 daughter cells, all right? And each one of these that you see my cursor pointing at will then go on and produce 2 more. So it's always going to be an even number. Now, there are some terminologies here that I want to reiterate, and you see me labeling them with a little bit of definition over and over again, because I know that could be confusing to you. So the blastula is this particular structure that you see here that consists of 128 cells. The interior of it, all right, it's filled with fluid. That fluid-filled cavity is known as a blastocele. All right, notice the differences in the vocabulary. And then as the embryo continues to grow and develop, it will form what is known as a gastrula, which is what you see down here. The gastrula is characterized by dramatic restructuring that results from cell migration. Essentially what happens now is that some of these cells begin to move into the inner layers, forming different layers of the primitive body part. And that's also where the cell begins to differentiate, meaning they become specialized. I'll talk more about that later on in this lecture. And you begin to form tissues, which then amass together to form specific body organs in a process known as organogenesis. All right. Now, here's something that I thought would be interesting to many of you, even though I'm kind of jumping ahead. You see the bottom of the gastrula right here, known as a blastopore. A pore, as you know, is an opening. That actually becomes our anus. And then there's a new opening that will be formed that gives rise to the mouth as we know it. All right. So now, what does cleavage actually look like? I find this topic particularly exciting, largely because we're very lucky. We have this technology where we can look at real life photographs. So take a look at this. Here is the pronuclei, as you have seen in some of the early video clips that I had recommended to you. I hope you can remember in one of the video clips in the previous lecture, you have the egg cell that have the awaiting pronuclei with the 23 chromosomes. And then as soon as a sperm cell comes in and the head penetrates the egg cell, it releases its own pronuclei that contains the other set of 23 chromosomes. Well, this is the fusion of it under a microscope, all right? That's what it looks like. Then the cell begins to undergo cell division. So one giant cell becomes 2 separate cells. Each one of these separate cells then go on to form 4 separate cells, and then each one of these carries on the process again, known as cleavage, where each of these cells produces 2 more cells, so they keep doubling at each of these subsequent stages. And this is the 28 cell stage, all right? Now, notice again, the overall circumference of these cells of the sphere remains the same, but the size of the individual cell, you see this one, how small it is compared to this one, right? It's much smaller. That's what's going on because the original size of this cell is way too big compared to a normal human body cell. So let's take a look at this. This is how the embryo develops with regard to, with respect to where it actually happens inside the woman's uterus. So here the ovary releases the egg cell, the waiting egg cell for the sperm. The sperm comes in, fertilizes the egg, then it forms into a zygote, then it begins to undergo the pairing, and then it starts to undergo mitosis, right? So from day 1 to day 2, mitosis occur, all right? Now, if you follow my cursor, you will notice how the cell doubles, and then it doesn't take that many days before it begins the process of implantation. This, the blastocyst implanting, by the way, you may have seen the word blastula. Blastula is a general term that refers to this particular structure in animals. Blastocyst specifically refers to mammals. If at any point you get confused, you can think of them as being very similarly to be just a mass of cells with the fluid-filled cavity known as the blastocele inside. So essentially, this is where it comes close. When the blastocyst is formed, then implantation begins to occur. That's where the embryo embeds itself into the mother's uterus lining, known as the endometrium. This is a special area that gets thickened and strengthens by the progesterone, and that's where the baby's going to grow for the next 9 months. I particularly find this very fascinating, perhaps because as a woman, I've experienced this process, and thinking about what the odds are against a sperm that can actually fertilize one egg cell that can still remain healthy at the time when I became pregnant, it is just an amazing process. If you think about all the odds that both the egg cell has to be able to defy over the years that I have been a grown adult, and also for sperm cell to find its way, not only to find the egg, but also not to fertilize the wrong object, as you have seen in one of the video clips. Sometimes they just fertilize any first round object that they come across. All right, let's move on. So in a schematic diagram format, this is kind of what it looks like. Now, the main thing that I just kind of want to reiterate and clarify for you, it's a lot of these terminologies that you come across. You may come across the word blastomere, by the way, and it refers to the 2-cell stage at this particular step. And essentially, what you're really looking at across all of these stages that my cursor is moving to, it's all mitosis that you're looking at. Now, the blastula stage then goes into gastrulation stage. Now, there is something called early gastrula. Early gastrula is when the cell already has made about 200 to 300 cells. Notice how tiny in real life the measurement would be. If you can actually take a ruler and try and measure it, it's extremely hard. You have to put it under a microscope. But this is the process when different layers of our body parts are beginning to form. We call this process of indentation invagination. So the layer right here of cells begin to invaginate into the embryo, and it starts the specialization process where organs and tissues can begin to form. When they are formed, they form different layers. There is one area here. You see the cavity? This looks like it's pointing to nothing, but actually it's pointing to a cavity. And keep in mind, these are fixed images, so it's hard for us to imagine, which is why I've also included live video clips for you to see it, how it occurs in other organisms, because it is so fascinating. This area called the archenteron becomes the digestive cavity inside our digestive tract. How cool is that? And then this inner layer right here, these lining of cells becomes the endoderm. Then you also have the ectoderm. Now, those of you who are really sharp and remember the material, you may notice the mesoderm, it's not labeled in here. That's because it hasn't been formed yet. All right, so now here's another visual aid that I figure would be helpful. hopefully not to, it's not intended to confuse you. If it's beginning to confuse you, maybe skip over one of these slides. All right, so here, after cleavage occurs at days 4 and 5, you have about 16 to 32 cells in a structure known as a morula. Then it forms a blastocyst because a lot more cells will then be formed. And one of the key things that happens here is that it begins to move some of these cells that used to be out here. They begin to form inside, it becoming what is known as the inner cell mass. Now, why is that important? Because this outer cell layer that you see right here, that forms the blastula, in mammals called a blastocyst, it forms the future chorion, which is necessary for the implantation of the baby onto the mother's placenta. That's what this is. This is one of the extra embryonic membranes that you have read about. Now, these inner cell mass, it's actually what's going to become our body organs and body tissues. And later on, in a few more slides, you will learn that these inner cell mass are really special. They are what we call embryonic stem cells. These embryonic stem cells have the capability of forming any body parts and body organs. We say that these are totipotent stem cells. And this is the beginning stage where the implantation occurs right at the mother's uterus, called the endometrium, right? Now, if you want to compare, just in case you're a little bit confused, and I know I'm giving you a lot of terminologies, look, here is the outer membrane in the blastocyst, and it forms the future chorion. This is what the chorion looks like. This is the part where chorionic villus sampling technique occurs. That's where you can take a sample of the baby cells to see if the baby would be developing normally, if it has the right number of chromosomes that we call karyotyping, remember? Okay, now, the other thing is that the outer membrane on some of these parts will also be used for forming the placenta. That is this portion of the embryo, this is the reason why I chose to give you one of these later developmental steps so that you can see what it eventually becomes. It is very hard to imagine how it actually folds and how it all occurs, but it is fascinating. Now, if you take a look at the late blastocyst phase, this is where the future embryo begins to form on the inside of the inner cell mass. You will notice that there is a formation of the amnion that encases the amniotic fluids, and also the area where the yolk sac would remain, that eventually will then contribute to the functions of the umbilical cord. All right, let's carry on. So here, what I want to show you is how the blastocyst invaginates. So you follow where the arrow is, it shows the process of invagination. Basically, it starts to form folds inside of the embryo, and then it forms all of these different layers, as I had mentioned earlier. Sometimes I find that when I can project a larger image on the screen, it would help your eyes visualize all of this. Now, one of the techniques that I learned when I was a student is to color code each one of these different layers in a different color on my note sheet, or when I draw it, because this way visually, it's more attractive and easier to encode the information, especially if you're trying to prepare for an exam. All right, so let's carry on and look at what happens next. So what happens is once the blastula stage proceeds into the early gastrula phase, not only does the germ layers begin to fold, which are the embryonic cell layers, they begin to fold inward. If you trace the arrow, that's what I'm trying to show you here. But they form the different layers, which becomes the different organs. So what's special about each of these cells such that you can get these different organs? Well, here it is. I love this particular slide because it's easy to follow and it basically summarizes almost everything you kind of need to know. So in this particular diagram, in the early stages, when the cell is undergoing cleavage, which is mitosis, all of these cells are said to be totipotent stem cells. What does that mean? Totipotent refers to the fact that the cell has total potential. They can actually form a whole organism. As a matter of fact, if you were to physically take the blastocyst and at the earlier enough stages, and you separate the cells, you can actually form artificial twins. And each one of those physical structures can then develop into two functional organisms. This type of experimentation has actually been done. All right, then what happens as the cell proceeds to go on to be forming a blastula? Um, then the blastula, in this particular case, has the inner cell mass. The inner cell mass is what we say that they have pluripotent stem cells. What does that mean? That means these particular cells have the capability of forming many other cell types in large categories of different functions. And they have the ability to sustain this replication among themselves. These are some of the cells that can then further later on become multipotent stem cells. What's a good example of multipotent stem cell? Like blood stem cells. So if you have a blood stem cell, those blood stem cells that are multipotent, which means it has a capability of reproducing itself for an extended period of time, they can also form highly specialized cells, such as red blood cells and white blood cells. Now, unipotent stem cells, as you may already have figured out, they are highly specialized cells, like specifically a B cell, a helper T cell, a dendritic cell, a neutrophil, a specific type of red blood cell. They cannot go back and they cannot change into the pluripotent stem cells because they have a highly designated function. So that's how the body is formed. Now, the embryonic stem cells in particular, which are the inner mass cells, are of particular interest to scientists because if you think about the function of these cells, the fact that they have this pluripotency in them, you can use it for repairing tissues and body organs. And that's where a lot of the research is being pushed. Let's take a look at it some more. So what do each one of these layers then form? All right, we've mentioned these ectoderm, endoderm, mesoderm. So this is one of those things that are very commonly tested. So this is one of those diagrams that I would like you to carefully label for yourself, but don't try to copy the slide. Just write down some of the key terms. The ectoderm tends to form the outer layer of the skin. The endoderm will then form the lining of our gut, our liver, and our lungs. Then the neuroectoderm, which is the midline on the top, will form our nervous system. Following downwards, the mesoderm, which is the reddish or salmon color area, will form our muscles, bones, kidneys, blood, gonads, and connective tissues. So these are some of the parts that you need to keep in mind, having some examples. All right, now in your textbook, there is a diagram similar to this one. This is a slide from a very, very old textbook, but nonetheless, this is still the same. So figure out a way in which you can understand some of these things. All right, ecto means the outer layer, meso means the middle layer, and endo means the inner layer. All right, now here is a footage of an actual frog undergoing cell differentiation from the blastula stage to gastrula, and you can actually see it moving. You can try to speed up the video and just watch how it happens and just take a bit of notes and then use that to connect to some of the concepts I mentioned earlier, all right? And I will give you this link separately underneath my lecture notes slide on YouTube. Okay, so how do body cells develop specialized functions as the embryonic cells are formed? All right, I gave you this particular video from Bozeman Science. He actually has two of them. They're very, very similar. So just follow the worksheet, answer the questions. Some of the diagrams on the worksheet are similar to the lecture note sheet, so you can use the information from this lecture to supplement your work, all right? So now, where has research gone with the studies of stem cells, which many of you I know are highly fascinated by? Well, not too long ago, back in 2012, a scientist by the name of Shinya Yamanaka from Japan, from Kyoto University, has figured out a way to convert a specialized types of cell, like specific adult bone marrow cells, skin cord blood cells, deciduous T cells. You take those multipotent stem cells, which has started to become specialized, you turn them back to form what we know as induced pluripotent stem cells, such that they regain somehow the ability or stimulate them such that they somehow can express those genes that has been turned off to work again, so that you can get it to form very specific body tissues and body organs for repair and for hopefully one day organ transplant. So he has gotten a Nobel Prize in 2012 for the success of his work. And you may want to look him up and take a look at it too. Now, here is a summary slide that I'd like to help you kind of encapsulate all of the different things that I have mentioned, because there's quite a bit that I have shared with you. Totipotent stem cells are special because they have the ability to form 3 germ layers, but not just that. I told you earlier that totipotent stem cells can form a whole organism. In order for you to form a whole organism, the organism must be able to produce the extra embryonic tissues or placental cells. Those are the areas like the chorion, the yolk sac, the allantois, all of those parts has to be there in order for this to work. Now, the pluripotent stem cells, what's special about them is they also have the ability to produce 3 germ layers, as I have shown you, but what they cannot do is that they cannot produce the extra embryonic tissues or placental cells. The multipotency stem cells are specialized cells that have very, very limited ability to self-renew, and they tend to be committed to lineage-specific differentiation, meaning that once they are designated to form particular types of body tissues or body organs, they will make those few parts that they are formed to do. But then somatic cells, on the other hand, are what we know as specialized body cells. But the one thing that they cannot do, not only can they not form embryonic tissues, as you already know, the extra embryonic tissues, but they also cannot be replicated too many times before they're worn out and they will be taken out by the body. So what are some of the extra embryonic membranes and what is their function? For the most part, they are very similar between a chick and a human. However, there are a few differences that I want to point out. The chorion that you find inside a chick, it's mainly used for gas exchange. For humans, it's used for gas exchange and also nutrient and waste exchange as well. The amnion is actually the same in both cases. Both organisms uses this to encase the amniotic fluid, which is indicated by the blue color that is shown here. The yolk sac that you see in the humans compared to the chicken is used a little bit differently. The yolk sac in the chick is used to store yolk as food for the growing embryo. But the yolk sac in human, it's mainly used for absorbing nutrients from the mother, forming blood cells, and also contributes to the function of the umbilical cord. Now, the allantois is also a little bit different. While both animals would use it for storing metabolic waste, in the case of chick, it's mostly urine, but in humans, it's also connected to the umbilical cord. But on the chick's side, it's also used as a respiratory surface. So there are a few things that are different between them, so that I wanted to highlight for you and also show you where they have things that are quite similar in terms of function. This particular video clip I have found by accident. When I first took a look at it, you can just see from the brownish color background, you know this was filmed a long, long time ago. As a matter of fact, I think it was produced in 1967. But what was so cool, such that I couldn't resist but to show you, is the fact that they are able to show a developing chick with the heart beating at various stages. I don't often come across video footage like this, but this one, it's really worth taking your time to look at it. However, I know time is limited, so I'm making this an optional activity if you choose. This is one of those old films that is not exactly on YouTube. You have to go back to the university website it's published by Indiana University. Go take a look when you have some extra time. Now you cannot, I don't think you can speed this up, but if you can, that'll be fun too. There isn't much narration. And what the scientist does is that they use a tweezer to lift up various parts of the body of the growing chick so that you can see when the tissues are still attached to each other, when have they differentiated enough such that not only can you see where they're different, but you can actually lift up a whole piece of tissue that you know are structurally sound and and has integrity. So that's why I wanted to show it to you. Now, this one, I definitely want you to see. Why? We have figured out a way to track exactly where do cells grow step by step over time in an actual organism. This is one of those latest findings that I have discovered only 2 years ago, and it was published, and it's super cool. So go to the link. It only takes you about 1 minute to take a look at, and the quality is really high. It's a very strong contrast to the previous one that I have just recommended for you to take a look at it too. All right, so how is embryonic development regulated? Well, you probably have read about the homeobox genes. What they are is essentially a large set of genetic material that can determine the basic structure and orientation of the organism, meaning where do you place the head, the arms, the limbs, the tail, or where the anus or the mouth would be. They literally direct the development and placement of specific body parts in order, in sequential order, in an animal's body. And this was discovered in part by using a type of fruit fly known as Drosophila melanogaster. For many of you, you may have already seen this particular fly. Sometimes you may even seen it hovering somewhere near the deli, especially where there are fruits, which is why they're called fruit flies, okay? But what's interesting here is that we have a very clear image on how the sequence of these genes are positioned in the chromosome and how that actually translates directly to the exact body parts that you can map out. Take a look at this, all right? Each one of these colors that you see on this image cross matches with the gene. So for example, you see where the head is colored in red, that is actually regulated by this gene that's positioned right here on the chromosome. And then you see where the mouth is right in this part, that's correlated to this part. Now you look at the yellow region, the next region over, T1, it's cross-linked to, well, not cross-linked, but this is regulated by this gene that's positioned right here. Certainly the color code, it's just an artist's rendition. But what's really cool is that you can see how the sequence, the positioning, the order of these genes directly corresponds to each of these sections of the body in the fruit fly. That is just phenomenal. You don't always see this. A lot of our genes are distributed throughout our 46 chromosomes, and they're not necessarily positioned this way. But what we do find that is extremely interesting, let me just jump ahead onto this slide for a moment, is that we now know these homeobox genes are not limited to Drosophila flies. You can also find them in mouse and humans as well. And it is also one of these special ways in which evolutionary biologists can use it to track and trace the changes in our evolutionary tree to form our cladogram, so we can see which organisms are more closely related to each other on the basis of biochemistry. And that's why I wanted you to show you this particular slide. So what are some of these genes? They essentially are made up of small segments of DNA, approximately 180 nucleotides, all right? Now, how do I know 180 nucleotides with code for 60 amino acids? Later on, when we get into molecular genetics, you will learn. You remember the nitrogen bases, adenosine, cytosine, thymine, guanine, A, C, T, G, those alphabets that you would see designated inside a double helix on a chromosome? Every 3 of these bases will code for 1 amino acid. So if you have a chain of 180 amino acids divided by 3 that is necessary for coding 1 amino acid, that's how scientists derive that it would form 60 amino acids as a consequence of that particular segment. These particular genes would encode proteins that serve regulatory purpose, like regulating transcription. For example, when do you copy certain segments of DNA such that you can form specific body parts or structures or molecules? And then these genes, most of these genes are organized sequentially on the chromosome, just like the way I described it earlier, so that the order of these genes would actually dictate the expression of those body parts in a real organism. In other words, if you were to go into, if we were somehow able to have a laboratory where we can play around by shifting and moving these genes, you will then be able to create, or, well, yes, you will be creating an organism that could be vastly mutated, depending upon what you have done and how much you have shifted the order and sequence of these genes. All right. Here is another example of a short video clip. This one you can also speed up. This is just for fun, so you can see why this is so fascinating to me and also to many of the scientists. What this does is that it shows the cell differentiation of a salamander embryo. I know some of you guys are animal lovers, and nothing speaks louder than words sometimes than having a nice visual. So take a look at this at your own time and see how you can tie in the things I've just talked about in the lecture along with what you see here. All right. So what happens during human embryonic development? Well, there are a couple of things. All right. At week 3, you see the formation of the amnion. It's clearly distinguished. Then you also have the amniotic cavity that is left over right here. You have the allantois that is formed right here. You have the beginning of the head fold. All right. That's in this region. And then you have the yolk sac. And then the chorion, which is right here, that's formed from the baby's body. Again, this is the area, usually it's down here in this area where the obstetrician will then extract a little bit of the cells for karyotyping to determine normalcy of a baby. All right. At week 4, you see the baby continues to grow, the enlargement of the embryo itself. You begin to see the tail fold. All right. And what you also see is the head fold has enlarged over time, and the egg and the yolk sac is reducing in size as well. Now, the other thing that is also really cool to see is that in retrospect to the positioning of the uterus, you will notice how the uterus lining has thickened over time. And there are a lot more vasculature of these blood vessels that you see here that are slowly to be ingrained into the mother's body. But remember, the blood of the baby and the mother actually do not mix. All right. Now, here is another close-up visual that I thought you might appreciate. It is a little bit complex. What you're looking at right here is that this is the umbilical cord, this whole section right there. And on this umbilical cord, you have arteries, which are designated right here, and then you also have veins that are there. And what begins to happen is that you have an exchange of material through these particular areas across each other. All right, you have the presence of the pool of maternal blood that is positioned right there. But remember, the blood on the mother's body actually does not go into the embryo itself, which is why both the baby and the mother are able to have different kinds of blood type. All right, so what is the function of a placenta? Well, the placenta is actually formed from both the chorion of the embryo and the endometrium of the mother. It contains a vast amount of capillaries that forms the inside of it, and it is mainly used for the diffusion of gases like CO2 and oxygen, and nutrient and waste exchange to keep the baby alive. And the mother is facilitating this whole process. The umbilical cord, the umbilical arteries, it's used for carrying deoxygenated blood from the fetus to the placenta, meaning you take out the CO2 from the baby and it goes into the mother's body. And the umbilical veins will then carry the blood, the oxygenated blood from the mother's body into the fetus so that the baby has oxygenated blood. The placenta can also secrete quite a bit of hormone that is used for exchanges of materials between the mother and child. Now, some things can be quite harmful. For example, if the mother were to intake a lot of drugs or alcohol, it can severely impair the growing fetus itself. Here is an image of the differences between a normal brain of a normal fetus, where the pregnant woman, it's not taking anything harmful or toxic that could impede the baby's growth, versus a woman who is taking in significant amounts of alcohol during the time of pregnancy. And some of the effects of this particular scenario is that it can lead to what is known as fetal alcohol syndrome. In fetal alcohol syndrome, the baby would result in very distinctive characteristics, such as a smaller brain called microcephaly. It can actually, the baby can also have much thinner lips that are characteristics of this of FAS. But some things that are not so obvious includes being mentally challenged. And the baby, as it grows and develops after it is born, will suffer from malformations of the skeletal system and major body organs. There's also inhibited growth, complications with the central nervous system, poor motor skills, and difficulty with learning and memory and social interaction, all of which, and this list is not even a comprehensive list of the degrees of impact from fetal alcohol syndrome. Here are some of the images of the growing, developing fetus that you can see. The suggestions on arm butt, I always find that to be really cute, fascinating, knowing that that's one of the first things that we reach for someone when we get to know them the first time is their hand. This is what the baby looks like at 7 weeks. Notice how transparent the baby's body is. And then this is at the 8th week. And that's the end of my lecture for today. I hope you enjoyed it, and take care, everyone.